I'll just wait a minute while people cycle in. Okay. It looks like we're recording. Welcome to Synaptic Connections, part of the undergraduate research symposium here at the University of Oregon. We are going to hear from a handful of outstandingly awesome undergraduate researchers, 10-minute, 15-minute presentations each. During those presentations, please, strongly, I encourage you to ask questions throughout. Do that via the Q&A or chat function that you have. And then after all four presenters finish, we will have a panel discussion. So this should be very fun. As always, please be courteous, polite. These presenters are, the top of their field when it comes to this kind of undergraduate research, and they are outstanding people. So please give them the respect that you would like to receive as well. Okay, so first up, I believe, is Isabelle. And she is going to start off our session for us. So please feel free to share your screen.
There we go. Hello everybody from wherever you are. My name is Isabelle Cullen. I'm a member of the SMEAR Lab in the Institute of Neuroscience here at the University of Oregon. And today I will be talking about my thesis project called Active Olfactomotor Responses in Head-Fixed Mice. So we will just be going through kind of this like a scientific paper. So first we'll go through some background and then into the research question, the implications of the research, methods, conclusions, future direction, and finally a summary. So for a little bit of background, the SMEAR Lab, we study olfaction, which is your sense of smell. And honestly, it's one of the most underrated senses that we have because it's important for a lot of different things, both in animals and humans. It can help us identify social cues, friend or foe, identify romantic partners, locate food, and even danger. But what is olfactomotor behaviors, as I said in the title? So olfactomotor behaviors are any kind of movement for the purpose of olfaction. So whether that be like the movement of your nose or contracting the muscles for inhalation. And they've actually been implicated in a variety of conditions such as Parkinson's, schizophrenia, Alzheimer's, and autism spectrum disorder, which is the main focus of my research. So autism spectrum disorder is a neurodevelopmental condition that is characterized by deficits in social and communication skills, narrowed interest, and repetitive behaviors, but also some sensory processing. So for example, my brother Davis has autism, and for him, this shows up in a variety of ways, whether that be flapping hands at times or repeating words to himself, or if he's unable to hold the natural back and forth of a conversation or maintain eye contact. And these are some of the more obvious symptoms of autism, but like I said, they also have deficits in sensory processing, and this is one of the more subtle, nuanced ones. And this actually also shows up in olfaction itself. So one of the papers that has actually inspired our work is a paper from Rosenkrantz in the Weizmann Institute in Israel. So normally when you're exposed to attractive odors such as roses, soap, or cookies, you actually take in a larger inhalation. So you take in a larger volume of air. Well, when you smell something aversive like trash or rotting food, you actually take in a smaller volume of air. And this is what typically developing children and neurotypical people do when they encounter these kinds of odors. So they wanted to see, do children with autism perform similarly to typically developing neurotypical people? And they found something really interesting. So individuals with ASD actually respond to aversive odors differently. So you can see in this figure C over here, this is kind of the graph that I just showed you of what neurotypical people will do when they smell pleasant and unpleasant odors. But in people with autism, there's actually no significant difference between attractive and aversive odors in terms of the volume that they inhale. But what's interesting though, is as an odor becomes more pleasant, typically developing children and neurotypical people, actually they'll rate it higher as a pleasant odor, but also increase their inhalation like I've shown you. But for people with autism, while they do say it's becoming more pleasant, they're not modulating their sniffing behavior. So that leads us to question, what is the neural mechanism that's causing this behavior to occur? So that is exactly what my research is dealing with. So for our project, we are actually continuing this in mice. So I'm sure you're curious, well, why continue this in mice instead of humans? So we're doing this for a variety of reasons. First being, mice have a really strong sense of olfaction. They actually have a lot more genes encoded in their genome for their olfactory bulb than humans. And they also exhibit a lot of olfactomotor behaviors. So you can see in this graph over here, this is measuring nose movement side to side. It's called yaw. Velocity, so the speed of the nose, both within trials and between trials. So when they're actually engaged in an olfactory task, they actually move their nose a lot, especially at the onset of odor, you can see over here. But when they're in between trials, you're actually seeing almost no movement in the nose. And this is kind of how it looks when they're actually engaged in the trial. And this is taken from one of our other papers that we recently published in eLife. So we're able to use, this is a great model organism to measure active sensing. And we can see this super well and measure it really well. But in addition, we've also, in the last 20 years, scientists have been able to identify different related genes to autism. And if you knock those out, so essentially where the term knockout comes from is just you cut out the gene entirely so the mouse doesn't produce the protein. And in doing that, you can actually produce a lot of autistic-like behaviors in mice. So for this project, we'll also be using autism mouse models, and the one specifically we'll be using is contactin-associated protein like 2, or CNTNAP2 mice that you'll hear me refer to. And they've actually been shown to have, to be less sociable, have decreased vocalizations, and show increased grooming behaviors. But we're specifically using these not only for these similar behaviors to autism, but also they've been shown to have olfactory deficits anatomically in terms of synaptic connections within the olfactory bulb, but also behaviorally. So how we're conducting this project is a couple of steps. So first, we're establishing that wild type mice do even perform a similar olfactomotor modulation, similar to typically developing children, as I showed you in Rosencrantz. And once we've established that, we can repeat the same paradigm in autistic mouse models or the CNT-NAP mice to see if this is even altered. And then we eventually hope to expand this experiment from a head fixed paradigm to a freely moving paradigm to see if these differ, and also record electrophysiology from the nose to see how the neural signals are actually going. So how we do this is a couple of things. First, we implant all the mice with a small device called a thermistor. And this is just a small instrument that measures temperatures and changes in temperature. And we use this as a proxy for breathing. So you can see that inhalations tend to be cooler and exhalations tend to be warmer. So we can use that as a great proxy for breathing. And then all mice are implanted with a head bar to keep them stable and not move around during the task. So once they've recovered from surgery, we expose them to several different odorants. And for aversive odorant, we're using 2-methylbutyric acid. For attractive odorant, we use 2-phenylethanol. For a neutral odorant, kind of in the middle of everything, we use alpha-pinene. And then for a control, we're using just clean air. And while we're exposing them to these different odorants for small intervals, we're also recording their orofacial movements. So essentially like the movement of the face and the nose while they're engaged in this task using 2 Raspberry Pi cameras, which you can see here and here. So far we've been able to conduct this on a couple of mice, but our analysis is only based on 2 mice at the moment. So the olfactory motor behaviors we've looked at so far are the inhalation patterns and the nose movements. So first, for the inhalation patterns that we've seen so far, we decided to look at sniffs per second because mice are obviously a lot smaller than we are and breathe more quickly. So this may give us a better idea of how they actually respond to these odors. So you can see here that the attractive odor noted in blue is actually upon like first inhalation, they actually have a way higher sniff rate for the attractive odorant than they do the aversive, which is a 2-methylbutyric acid, or the neutral, or even the blank. So this is really interesting and leads us to see, okay, obviously there is something going on here. And like I said, this is kind of based on a small sample size. So we will need to continue to repeat this in other mice to see if this same effect still stands. But for the orofacial movements, as you can see, the mice, so let me play the video first. This is actually a video taken from one of the trials. As you can see, the mice actually move their nose a lot in response to these odors. And we can actually track this positioning using AI system called Deep Lab Cut, which allows us to place dots on spots on the animal that we want to track. So we'll use the positioning of these dots later to track how much the nose moves side to side and laterally, and see if there's any kind of correlation between the orofacial movements or olfactomotor movements between that and the odor. So what we've established so far is that wild type mice perform more sniffs per second to the attractive odor than the aversive, neutral, or control. And further analysis is needed for the nose olfactomotor movements, which I showed you in the video, to see the responses to the odor. Do they move a lot? Do they not? But ultimately, we do need to collect a larger sample size in order to make definitive conclusions, because this is based on a sample size of 2. Some of the future directions we hope to go with this project, like I said, is using a freely moving paradigm. So when I said head fixed, they're literally put in a little box and just stabilized like this. So why we want to look at this in a freely moving paradigm is obviously when you're in nature, you're moving around, you're doing different things, you're not stuck in one position. So this is interesting to see, will the neurons themselves actually fire differently if they're freely moving rather than head fixed? And we'll do this by repeating a similar thing with using the thermistor, but also adding electrophysiology little doodads to record neuron signals within the olfactory bulb. And we have significant promising data to suggest that this actually might be true. Our lab has shown recently that in 2 different cells, so this is one cell and a second cell within the olfactory bulb, these 2 different cells actually have very different temporal patterns of firing when they're head fixed in the top row, when they're versus when they're freely moving. So you can see down here, these 2 just look very, very different. And this is really great because it tells us that maybe the olfactory bulb and the neural firing is maybe modulated by movement or behavioral states. And this may ultimately affect olfactomotor behaviors. So we might be studying a lot more in the future. But that is still to come in the future, and hopefully future undergrads will be able to continue and see that work, which I'm very excited for. So to sum up everything, our results, or our study, is motivated by the results of the Rosencrantz paper, which found that children with autism do not modulate their sniffing in response to aversive odors. So we're hoping to gain a better understanding of the neural basis of ASD and olfactomotor behaviors through this research. And like I said, and I've shown you so far, we are studying this in mice, and we plan to, and have done so far, to establish just a baseline of, do they take similar olfactomotor behaviors to typically developing children using wild type mice. And then eventually we plan to repeat the same paradigm in CNTNAP2 mice, or the autism model mouse, to see if their olfactomotor behaviors do differ. But with that, I'd really like to acknowledge quite a few people. First off, Jared and Avinash for all of the work that they've done in my absence during COVID, because I've been remote this entire time and them setting up the system and doing all the coding. It's just been phenomenal to both work with them and have their expertise in this, but also Matt for all of his guidance and everything that I've done over the last 2 years, but also some funding that I've had, such as a Peter O'Day Fellowship in Europe. Obviously the whole Smear Lab, because they all contribute to this in multiple different ways. The Robert D. Clark Honors College for allowing me to continue this thesis, but also the Department of Neuroscience for this to also be my honors thesis as well. Thank you so much. And now I will open it up for questions.
So we're actually going to take questions at the end, but please, please, please post them into the chat, into the Q&A throughout the entire thing, and we will have a panel discussion afterwards. Cool. Thanks, Isabelle. Everybody give like a round of applause. I know it's hard digitally. Okay, so next up we have Alyssa, who's going to tell us about the comparison of V1 activities between mice raised in enriched versus standard housing conditions. Take it away.
Hello, everyone. Let me just get this set up. Perfect. Okay, so my name is Alyssa Fuentez, and I will be presenting my McNair's research project that I conducted with the Neal Lab that is titled, Does a Stimulating Environment During Development Affect Activity of Visual Cortex in the Mouse? So pediatricians and psychiatrists suggest that parents provide a stimulating environment for their children. That's because visual, tactile, and social stimuli are great for wiring the developing brain. So if we know this, then does this apply to some of the model organisms used in research? These experiments were conducted in a vision lab. So we were interested in how the standard housing conditions for adult mice in vision studies, how that housing impacts visual development. So briefly looking at the housing conditions on the left, we see that it mimics the nursery that lacks basic stimuli. So we designed our experiments to compare the impacts of that housing to those of mice raised in an enriched interactive environment. Before I get into the experiments, I'd like to go over some background information, and I will begin with an overview of the visual pathway. As light enters the eye, it will be sensed by specialized cells, and their job is to take that light stimulus and transform it into an electrical message that it can send to different locations in the brain. That's how the brain communicates. So what's also forming at this time is going to be what's called a retinotopic map. What that means is that that location has a map of whatever is in the visual field. So in this case, because we're in the retina and because the eye is looking at a tree, the retina has a map of the tree. And it's going to take that map and it's going to send it to different locations. First, it goes to DLGN, where basic processing occurs. However, for the most part, it takes that map and sends it to primary visual cortex, or V1 for short. So visual cortex as a whole is going to be the location of more complex visual processing. And it's able to do that because it's communicating closely with all of these surrounding higher order visual areas, or HVAs for short. So each of those surrounding areas, they have their own maps, and they're specialized to respond to specific features in those maps. The two key takeaways from this slide are going to be that V1 and the HVAs are the locations of complex visual processing, and a map is maintained throughout the visual pathway. Now let's look at spatial acuity and the visual scene in real time. I'm sure we've all seen these tests where you cover one eye and are asked to read down as far as you can. That is testing what's called spatial acuity, which is the finest detail that can be detected. So when testing spatial acuity, we're able to get a benchmark for that organism's visual system condition. And an organism's spatial acuity is going to be dictated by the range of spatial frequencies that it can detect. So spatial frequency is defined as the pattern of light and darkness in an image. High spatial frequencies correspond to those fine details and sharp edges, whereas low spatial frequencies correspond to more global shape and form. So if an organism has a wide range of spatial frequencies it can see those spikes on the cactus because it has a high spatial acuity. Whereas if its range was more narrow, it would just see that global shape of the cactus. Now let's look at the visual scene in real time. This is a children's book series called Animorphs, and something that is characteristic of them is that if you flip the book pages, the silhouette of the main character becomes whatever creature it's going to become. In this case, it's a lizard. And I'm showing this video because I think it's a great way of illustrating temporal acuity and temporal frequency. So these are referring to movement in the visual scene. If we were to be moving these pages slowly, we would be able to see features of that as it moves a little bit more clearly. The silhouettes would be more clear. However, as the pages start flipping more rapidly, your brain is only getting snapshots of those images before they move. And the movement becomes so fast that what happens is those pages begin to blur. You begin to see one page and the main character becomes a lizard in this case. So by looking at and measuring an organism's temporals or responses to temporal and spatial frequencies, we're able to get an overall look at the quality of the visual system. Now let's look at the standard housing conditions for lab mice. As I stated before, these mice are raised in just the essentials, and that is not good because previous research shows that visual deprivation negatively impacted visual development. Now, reassessing this, not only do we see that the environment lacks visual stimuli, it lacks experience in general, which is huge for the visual system. Think about it and think about everything that you're looking at and imagine that all of that light is going to be like a pixel, and your brain is responsible for processing every single teeny tiny pixel of light. That is a lot of information to process. And then you add on the component that we live in a dynamic environment that's changing all the time. It's too much. So the brain, to increase efficiency, what it does is it makes informed guesses about the environment. And that's all based on experience. You can see that in this test. There are multiple components going on in this. However, because we have the experience of seeing a checkerboard and the white and dark contrasting blocks, we think that B would be lighter than A. However, they're actually the same color. So if we know that experience is essential for visual functioning and development, then why are the model organisms that we use to study research raised in environments that lack stimulation and experience? That is the scope of this research, which leads me to the research question, which is, how does enriched housing during development of the mouse affect visual cortical activity? We wanted our enriched housing as having cognitive, physical, visual, and social stimuli. So to achieve that, we had increased mother rearing time. There was a running wheel, a maze, an iPad that showed a slideshow of various images, and then mice were raised in cohorts to promote socialization. So to test our question, what we did was we first raised the mice in both housing conditions, and then we quantified the differences in cortical activity. So we're looking at brain activity here. And as I said, we're looking at the brain activity and responses to varying spatial and temporal frequencies. The way we're able to measure brain activity is we use these special mice whose brains, or their visual cortex more specifically, glows when it's active. And that's what you're seeing in B. It's an actual glowing mouse brain, which is pretty wild. But so we have these mice, and then we show them various movies and various images, and then we rapidly image the brain and look at how much it's glowing, because if it's glowing a lot, that means there's a lot of activity. So I want to show you some of the data that we collect in these experiments, but first I want to remind you of this. The idea that visual cortex, all of these areas have their own maps of the visual scene, and you are going to see that right now. So in the top left corner, that is what the mouse is looking at. And simultaneously, you're seeing these maps of activity moving across these different areas. So in that center, that large bar, that is going to be V1, and then all of these surrounding areas are the HPAs. It's pretty amazing. So to detail some of the stimuli we use, I have a picture of a patch grading. This is a movie, so it has movement in it, so it's testing both spatial and temporal frequency. And then we use a series of natural images to test the limits of the spatial frequencies. This is some of the data that we have gathered in response to the patch grading stimulus. So on the y-axis, we're looking at response magnitude. DF of F means difference in fluorescence. So essentially, is the brain glowing more? Because that means it's more active. And then on the x-axis, we have X position, and that's just referring to the position the circle is on the screen, 1, 2, 3, or 4. And in all three areas, we're seeing that the enriched cohort is responding more, so their brain is more active. However, when we look at their responses to various spatial and temporal frequencies, we're not seeing that significant difference. However, what we're also not seeing is a bell shape. So organisms, they cannot sense an infinite number of amount of spatial and temporal frequencies. There should be stark declines on both ends of each of these graphs. So this leads us to believe that we did not push the limits of their ranges. This is data from the natural images, and it's separated by area. Once again, we're looking at the difference of fluorescence, and we see that for all of these, they are significantly increased for the enrichment cohort, but we're seeing a larger difference in V1, which would make sense because that's primary visual cortex, so it's going to be receiving the most information. Finally, onto my favorite figure. This one is really interesting. So if we look at A first, we're looking at the picture of the cactus and then the mice response to that. And what we're seeing is that the enriched cohort apparently really likes this photo. There's a really big difference between the control and the enriched. However, when we look at their responses to the bird in B, there's not a significant difference between the control, which brings up many questions. Maybe there's textures at play. The bird that just may not have a lot of visual stimulation in the image. So we have a series of natural images. So there's a lot to dig into on this one. So to conclude, we found that all three areas were responding higher for the enrichment cohort. The response magnitudes vary between the areas, with B1 having the most significant difference in enriched response. Responses are image specific, and we need to further test the limits of spatial and temporal frequency in these mice. And with that, my presentation is finished, and I just want to acknowledge everyone who has helped me along the way in completing this project. Thank you.
Everybody give Alyssa a round of applause. And please, please, please be putting your questions into the chat. And we will pass them around to the panel at the end of the day. Okay. Next up is Katherine. And please correct me if I pronounce your name wrong. Who is going to talk about decoding the role of the auditory cortex during prey capture behavior in laboratory mice.
Yes. Hi.
Please take it away.
Let me get that bar moved and we can present. All right. So, all right. I don't know why it's at the very end. I apologize. Okay. So my name is Katherine Kaylegian, and I'm going to talk about decoding the role of the auditory cortex during prey capture behavior in laboratory mice. So prey capture is really just this fancy word we use for hunting. And we don't normally think of mice as hunters, but they are. And they hunt mostly insects, and they hunt using mostly auditory input. This is what we consider a dynamic two-bodied problem, because the mouse is moving in response to the cricket and the cricket is moving in response to the mouse. So this actually becomes quite a complex behavior to try and figure out what's really going on. So we'll get into that a little bit later about how we actually start breaking this behavior down into usable pieces so that we can get some sense of what's actually going on. But before we can do that, we note that from previous research, there are two possible pathways that auditory input can take for processing and outputting a usable behavior. The first pathway uses just the superior colliculus, which is an evolutionarily ancient structure present even in reptiles. The second processing pathway, auditory input, can go from the superior colliculus to the auditory cortex and then produce a usable behavior. And the auditory cortex is a much newer structure, and we don't really know what its purpose is, but it is hypothesized that the auditory cortex is used for differentiating or for processing information from the environment. So then we ask ourselves, what is the role of the auditory cortex during prey capture? And to answer this, we first need to figure out, are there quantifiable differences between the two processing pathways? So we start answering our questions by noticing that prey capture behavior can be broken into discrete states. So this is really how we start trying to figure out what's going on, because we notice that either the mouse knows where the cricket is or the mouse doesn't know where the cricket is. So if the mouse is not aware of the cricket, we call it search. If the mouse is aware of the cricket, we call it pursuit. And then we have kill or capture. And the mouse can't know where the cricket is and not know where the cricket is at the same time. So these discrete states lend themselves to using a hidden Markov model, which I will also talk about a little bit in a minute. But to get our data, we use video trials using infrared cameras in a dark arena to ensure that the mouse has the use of only the auditory cortex. And we use saline and lidocaine injections. The mice given the saline injections have the use of the auditory cortex, so their auditory information is going through the superior colliculus and the auditory cortex. And then we give lidocaine injections, and those mice only have the use of the superior colliculus. This is supposed to be a video, but it doesn't play, and I have no idea why, and I can't make it play. But this is really just to give you an idea of how simple our setup is. It really is just this circular arena with some aerial cameras and this mouse. And then we have a cricket launcher that's really kind of flinging crickets in one at a time. So we fling a cricket in, we turn the cameras on, we video, and we get something that looks like this. But this doesn't tell us anything about what is actually going on. So to get some information about what the mouse is actually doing as it is hunting the cricket, we use deep lab cut, which is a deep neural network, and we use a hidden Markov model. So deep lab cut does this really awesome thing where we can take about 40 frames out of all of our videos, label the mouse and the cricket or whatever bodies we're interested in with these little markers, and then we train deep lab cut overnight, and it puts these wonderful markers on all of our videos. But again, by itself, these markers don't really tell us anything. The cool thing that deep lab cut gives us is position data. So then using that position data, we use a hidden Markov model, which will take our videos and a set of variables we give it. Currently, so we've had at most 14 variables that we give our model. Right now, we are using a reduced model because we find that it gives cleaner states. Excuse me. So right now we are using mouse position, cricket position, mouse velocity, cricket velocity, and azimuth. And we define azimuth as the angle between the mouse and the cricket. So we give our hidden Markov model all of this information, and then it breaks our videos up into these states. And so you can see here where the state is listed with a K and the epoch number E. The state is really just an arbitrary number that the hidden Markov model uses to denote groupings of similar characteristics. So that means that hopefully state 3 has certain characteristics that each of these epochs shares. And then the epoch number just denotes where in our videos that this clip came from. So the first thing that I do is look at these videos and make sure that there's congruency within states. So I'm looking to see that most of the epochs, the mouse either knows where the cricket is or doesn't know where the cricket is. And if I see good congruency within the states, then I look at a clustered transition probability matrix. So this is a cool piece of data that our hidden Markov model gives us. And the transition probability is the probability of going from one state into the next. So if you look at the plot on the left, the x-axis has the current state. The next possible state that the mouse could transition to is on the y-axis. And this is just a heat map of possible transitions. This TPM has been clustered, which means that we're grouping by similar states. So we're taking all of the states that have similar characteristics and we're grouping them together. And when we do that, what we find is that the highest transition probabilities occur within these clusters, which means that if the mouse is in search, the mouse really wants to stay in search. And if the mouse is in pursuit, the mouse also wants to stay in pursuit. And then if you take a look at these clusters that are circled on the diagonal, this is pretty typical of what a good run looks like, where this cluster up in the top left is a pursuit cluster. So this means that all of these states that are circled with the big pink circle are all pursuit states. And so typically we'll have one bigger pursuit cluster and then a number of smaller clusters that are search or mixed, a mixed cluster having search and pursuit states in it. But our question right now is, are there differences between saline and lidocaine trials? So once I've looked at all of that and I can confirm that this is indeed a good run and that our model has indeed done what we hope it is doing, we look at the TPMs separated out by saline and lidocaine trials. And if you take a look at my arrows, hopefully you guys will be able to notice the differences in transition probabilities between saline and lidocaine trials. And we can start to make sense of this when we quantify these differences, which we do by taking the mean dwell time spent in search and in pursuit, and then separating that out by saline and lidocaine trials. And here, if you look at the plot on the right, you can see saline plotted in gray and lidocaine plotted in red. And what you see is there is a huge discrepancy in the dwell time when a mouse has the use of the auditory cortex versus when the mouse does not have the use of the auditory cortex, because this little gray bar here is more than half the height of the lidocaine bar. And even though the discrepancy in pursuit isn't as great, there still is a discrepancy, which means it takes the mouse less time when it has the use of the auditory cortex to actually capture the cricket than when the mouse does not have the use of auditory cortex. And so this is an agreement with our hypothesis that A1 cortical projections serve to help the mouse differentiate sounds from the complex environment to correctly orient towards an appetite of stimulus. Our next steps then, since I've talked about differentiating background noise, is to actually add in background noise to varying levels. And if our hypothesis is correct, we expect to see even more drastic differences in state dwell times between search and between like saline and lidocaine trials. And we also want to start taking physiology data with our behavior data, because now that we have a good idea of what's going on and how to break this behavior down, we want to know what neural circuitry is actually responsible for this behavior. And then I would like to thank everyone who has contributed to my research, and I'd like to thank you all for listening.
Again, everybody, round of applause. And once again, I'm going to keep harping. Please put your questions in the chat. We're going to get to them in a couple of minutes here. Next up is Amanda, and she's going to be talking to us about what you can see on her screen. So here we are.
Hello, my name is Amanda Linskens, and I'm an undergraduate research assistant at Doe Lab within the University of Oregon. My research mentor is Kristen Lee, and my project aims to better understand the molecular origins of the para-1 and Moonwalker descending neurons and their neural circuitry in Drosophila, also known as fruit flies. So neurons are very important for complex life, and the neurons specifically are important for eliciting certain behaviors, and in this case, such as movements. So like moving your hands or walking or throwing an object or something. And so in order for these neurons to elicit this assigned function, they need to be able to communicate with each other somehow. And so this is usually done through circuitries. So the way that circuits works is that a signal is first propagated in the cell body of the first neuron, and then the signal is transmitted down the axon of the first neuron to the dendrites of the second neuron. It then proceeds back to the cell body of the second neuron, where it is able to repeat this signal transduction over and over again until it reaches its desired location. That way it can display the proper behavior. So this means that any two neurons that are able to form this type of connection are considered to be in a circuitry with each other. So a lot of research has focused on how these individual neurons are able to develop and the molecular underpinnings that go into that development. However, not a lot of research has looked at how these two neurons know that they need to form this circuitry with each other and the molecular influences that go into that. So we decided to look at this using the Moonwalker descending neurons, also known as the MDNs, as they are able to form a circuit with the pair 1 neurons to control locomotion in both fruit fly larvae and adults. So we decided to focus on the Moonwalker descending neurons and the pair 1s because they are both very well characterized in the fruit fly brain in that they have a very well-defined morphology and function. So in this schematic here, we can see the central nervous system of 2 larval brains. And one is the MDNs and their morphology. So we can see 4 MDNs that are located very high up in the brain, and they have axons that descend all the way down into the VNC, which is where the premotor neurons live. With the pair 1s, they have 2 cell bodies located a bit further down in the brain, but also have axons that descend all the way down into the VNC. So the MDNs control backwards locomotion, and these are able to activate the pair 1s, which control pausing. So both of these neurons are able to activate premotor neurons in the VNC, which will then go on to activate the motor neurons. So like all other neurons, the MDNs and pair 1s are derived from neuroblasts, also known as neural stem cells in vertebrates. So in this schematic seen here, these large circles are the neuroblasts, and they are able to asymmetrically divide into these smaller circles, which are the neuronal progeny. So as the neuroblasts develop through time, they are able to develop through certain temporal windows that are marked by specific temporal transcription factor expression. So the first temporal transcription factor that is expressed is hunchback. And then as the neuroblast divides into the neural progeny, the progeny is able to inherit hunchback while the neuroblast moves on to crouple. And so these temporal transcription factors are very important for determining the neuron's fate and the way that it functions by controlling genes that are either activated or inhibited in those specific cells. So little research has focused on neural circuitry formation and maintenance. So how exactly do the temporal transcription factors lead to formation and maintenance in the neural circuitry? So we decided to first look at this by determining which temporal transcription factors the MDNs and pair 1s derive from. So we decided to do this using the GAL4 UAS system to be able to visualize these neurons. And so the way that the GAL4 UAS system works is that you have a neuron-specific enhancer that is only expressed in either the pair 1s or the MDNs. And so in specifically the pair 1s or the MDNs, this activates the expression of the GAL4 protein. The GAL4 protein can then go bind to the UAS binding site and activate the expression of the green fluorescent protein, also known as GFP. So GFP can then be stained for because it is membrane bound. And so the way this works is that we are able to dissect the larval brains, antibody stain for them, and then image them. And we are also able to antibody stain for these 4 different temporal transcription factors And so what we're looking for with these images is whether these temporal transcription factors overlap with the GFP staining. So the images for the MDNs can be seen here. So the GFP is staining the membranes of the MDNs, and then in magenta is the hunchback staining, and then the image over here is showing the composite image of the two different stains. And so here we can see that the MDNs are co-localizing with the hunchback, and this was even further supported by quantifying these overlaps. So on the y-axis, we can see the different temporal transcription factors that we tested for, along with the number of neurons that we saw. And then on the x-axis, we can see the percentage of the MDNs that co-localized with that temporal transcription factor. And here again, we can see that the temporal transcription factor hunchback strongly co-localizes with the MDNs. We were also able to see a similar co-localization with the pair ones, where again, GFP, instead of staining for MDNs, it's staining for the pair ones, and then in magenta is the hunchback. And here we can see that they are co-localizing. Again, we were able to quantify this and see that the hunchback strongly co-localized with the pair one neurons. So this means that both the pair ones and the MDNs are derived from the temporal transcription factor hunchback. So with knowing that hunchback is a temporal window that the pair ones derive from, we wanted to see what would happen if we were to knock down or overexpress hunchback in the pair ones. And so this again can be done through the GAL4 UAS system. So only in the pair ones, this system is going to be expressed. So the neuron specific enhancer is able to activate the GAL4, and along with activating GFP, it also is able to bind to this other UAS binding site somewhere along the DNA. And this activates either a hunchback RNAi, which is the knockdown of hunchback, or a hunchback transgene, which is the overexpression of hunchback. So again, we were able to dissect the larval brains and antibody stain, and then image these brains to see what changes in morphology could occur. So with the control here, we have the two cell bodies for the pair ones in green, which is the GFP staining. And then we also see the axons descending all the way down the side of the magenta neuropil, which is stained by the NC82. And the neuropil is just a very synapse-rich area. With the hunchback, we were still able to see the two cell bodies. However, the axons weren't able to descend down the side of the neuropil as much. With the hunchback overexpression, we still did see the axons descending down the side of the neuropil, but instead of two pair one neurons, we actually saw three. So these results were really interesting, especially since morphology tends to be related back to the function of these circuits. So we decided to look at the behavioral changes associated with the hunchback knockdown or overexpression. The problem with this is that we need to know when the pair ones are actually turning on. And so this can be done through an optogenetically activated cation channel. And so the way that this works is that in the presence of light, these pair ones will be able to turn on. And so the way that that is done is by treating the ATR group with all trans retinol, also known as ATR, and then a control group that is treated with vehicle instead. So the way that this works is when the red light is off, both of the pair ones are off in both the ATR and the vehicle group, and so they are both able to move forward. However, when the red light turns on, the pair ones are still off in the vehicle group, but the pair ones turn on in the ATR group, and they are able to elicit the pair ones function. We can also add in another key element to this, which is the hunchback knockdown and overexpression, to see how the behaviors change between the controls and these different experimental groups. So the results from that can be shown here. So on the x-axis, we have the time, and then on the y-axis, we have the speed of the larvae. So in blue is the ATR group, and then in white is the vehicle group. And when the red box is shown here, that's when the light turns on. And as we can see, the ATR group is able to slow down which shows that the pair ones are being turned on. And then as the red light turns off, the pair one or the ATR group is able to speed back up, showing that the pair ones have turned off. So this was a bit different in the hunchback knockdown. So while we still did see the pause here, this was a lot more accentuated compared to the ATR and the control. And we were also able to see that it took a bit longer for these larvae to speed back up again once the light turned off. And this slowdown right here can actually be looked at a bit further and shown that this is pretty significant in that the deacceleration of the hunchback knockdowns was a lot more emphasized than the ATR in the control. With the hunchback overexpression, again, we were able to see this slowdown again, and this was, again, a lot more accentuated in the hunchback overexpression. And we were also able to see that regardless of when this light turned off, immediately the hunchback overexpression larvae were able to start speeding back up again in the ATR group. And so we were again able to look at this slowdown and see if that was experimentally significant or not. And what we were able to see was that the hunchback overexpression was a lot more emphasized in its deacceleration when the light turned on compared to the control group. So some conclusions that we were able to draw from this. So the MDN and pair 1 neurons are derived from the same temporal transcription factor, hunchback, which could indicate that the neurons from similar developmental origins function together in a neural circuitry with each other. We were also able to determine that changing the amount of hunchback in pair 1 neurons affects morphology and behavior, and that new morphologies and behaviors suggest that normal pair 1 circuitry formation is altered by changing the molecular origins. So a few future directions. What we plan on doing next is performing the same hunchback knockdown and overexpression and looking at behavioral changes along with morphological changes, but this time in the MDNs with the larvae and the pair 1s and MDNs with the adult fruit flies. We also plan to perform neurite mapping of the pair 1s when hunchback is knocked down or overexpressed to better look at what exactly is changing with these circuitries to give this new behavior. So I'd like to thank all of you for listening to this, and a special thanks to everybody in the DOE lab, especially Senlin, Lorena, and Kristen for helping me with this project. And I'd also like to thank SPUR and the NIH for funding this research. Thank you.
Awesome. Again, please give a digital round of clapping for Amanda and all of our panelists here. So again, I'll introduce myself. I'm Pete Batzel. I work for the bioinformatics and genomics track, part of the Night Campus internship program. And I have no idea, like I've never interacted with sets of people that know about synapses or anything to do with that. So your comments will be more like better for conversation than mine. So please toss them in here, get them into the questions section, and we will we'll talk about them. So panelists, feel free to turn on your cameras and just hang out, and we'll have a couple of sets of discussions. We do have a couple of questions in the chat. Some are pointed, some are not. So we'll take them one by one. The first is, Isabelle, how would you be able to measure orofacial movements in non-head fixed experiments? So if their heads aren't fixed, is that what the implanted electrodes are for?
Yeah, that's a great question, Jessica. So we wouldn't necessarily be able to measure it from the side. However, with some of the experiments we run currently in our lab, we're able to use 3D tracking from above to look at the nose position relative to the head to the center of mass. So we can measure the angle movement of the nose, but we can't, see it, whether it's going up and down, side to side, and some of those finer movements. But the electrodes are more to look at the neural activity within the bulb rather than looking at the orofacial movements themselves of the nose.
I have a follow-up. So I'm a geneticist, I guess, at this point. So this is all based on live animals. Is there a way that we could get at some kind of genetic response to any kind of olfactory that's going through this?
Could you rephrase your question? I'm not sure I understand.
And I'm just wondering, I don't know, is there most of the time the olfactory research or regardless research that I've been studying is based on like young animals that we're genetically sequencing what we think is their olfactory system or their olfactory bulb or set of neurons. Is there a way to like stimulate that and then I don't know, ask genetic questions and see if we can.
No, I think I understand what you're saying. So we can actually use specific type of mice. I forget which ones they are, but they have specific tagging markers so that we can either do like viral injections with certain things that have like, it'll add like a fluorescent protein like Alyssa used in her experiments to activate like or look at the activity specifically of certain types of neurons or areas within the olfactory bulb. And we can use those in a variety of ways to look at that in live animals, and same even with just like the autism ones, we can take those genes specifically out and see how that affects the behavioral states of the animal if those genes aren't present.
This is like crazy cutting edge stuff. Things that we couldn't do like 5 years ago. Yeah, this is awesome. Okay, I'll switch to the next question. Alyssa, could finding a way to mimic the natural environment of non-lab mice be the optimal way to test how their visual acuities are influenced by lab conditions?
So that is a great question. And so I'm first going to respond with something funny. It was just the initial thing that popped up. And when we were showing the various natural images to the mouse, there's an image of a tiger. And I was like, isn't that going to elicit fear in the mouse? But the mice are from the lab. So that's not that relevant, but that is the first thing that I thought of, that we think of mice in this kind of natural environment, but they are in labs, so they're not used to that natural environment. However, to answer your question more specifically, I think that just the constraints of the housing for the mice, it is even just our enriched environment. It's a little bit laborious for cleaning and funding, and it's a bit difficult to upkeep. So when we think about the stimuli that like we incorporated, for example, a running wheel, if we wanted to have a natural environment that provided that physical activity for a mouse to run 30 minutes if it wanted to, that environment would not necessarily be able to, I mean, I'm sure it could be created, but realistically, I'm not sure we could do it. Also, for example, like we have a maze for cognitive stimulation, and thinking about where animals would get that in the natural environment, I imagine that would come more from unexpected events or various scenarios, and we can't necessarily mimic those types of events. So we designed our enrichment based off of some previous research that shows that has an enriched environment, like the increased mother rearing time has been shown to be beneficial. And we just wanted to make sure that we tried to hit as many aspects of stimulation and experience as we could.
Anybody have follow-up thoughts? So I can tell you that in lab-raised zebrafish, you know, if you put your hand above their tank, they all flock to it because they know that you're feeding them. And in wild zebrafish, if you put your hand anywhere near the tank, everybody scatters. And so, yeah, well, you know, lab environment is like very important for all of these things. Whether it matters for one specific assay or the next, I don't know, but at least that anyone is thinking about it is like 2 thumbs up. So outstanding. Cool. Okay, we have one more question. Amanda, how is the pausing behavior in the ATR group measured? Do we know if HB influences the morphology and maybe behavior of most other neurons, since it is a sort of precursor transcription factor?
Yeah, so that's a great question. I'll answer the first question first. So with the pausing behavior in the ATR groups and how that's measured, we actually use a specific instrument for that, and that's called a FIM table. And so we're able to put these fruit fly larvae onto a gel, and then we're able to analyze their movements and track that with a video, and then turn on the red light and then turn it off. So it's a pretty set time that we have to record them. And then what we do is we take the video and then we put it through a certain program that is used to FIM, and we're able to basically measure how fast these larvae are moving, and specifically with their velocity, also known as speed. And so we're then able to put that into a prism to then look at these, the different averages of the movement with these larvae. For the second question, we do know that hunchback is able to influence some of the morphology in some of the neurons, especially with the ones that hunchback is derived from. However, there's not too much research into how that specifically affects the circuitries. And the biggest point with hunchback is that since it is a temporal transcription factor, it's meant to activate and inhibit expression of other genes. So the best way that we can look at how hunchback influences the morphology and probably the behavior is actually by looking at how it influences genes of other transcription factors, such as identity transcription factors. And that's actually one of our next plans in looking at this, as to how exactly hunchback, when it is knocked down or overexpressed, how that exactly influences the expression of these specific identity transcription factors. However, I can say that with the MDNs, we have started looking at them with the hunchback knockdowns and overexpressions, especially with the morphology in the larval brain, and it does elicit a similar morphology where the axons are cut a bit shorter than what you would normally see in a controlled morphology. So I think that's really interesting. But again, most of the research into the hunchback and how that influences the morphology is mainly looking at the cell bodies.
Cool. Any comments or thoughts? It was kind of a specific question. Okay, well, I will, you know, please, you know, send us anything that you would like to ask, and we will make it, you know, straight in here. What I would like to know, just to open it up to the panel, is when I was an undergrad and then a graduate student, it was impossible to come up with a thesis statement, to come up with a thesis idea, I thought. So if anyone would like to talk to, how did you come up with your hypothesis?
I'm working off of data that a master's student started a couple years ago, and her project was titled "The Auditory Cortex is Not Needed for Prey Capture Behavior." And so then my project was just kind of based off of that, that if it's not needed, why does this processing pathway exist? But the project was really just kind of handed to me, and then I started doing the data analysis, so I didn't really get to make it up.
I think that still counts. Taking it up is making it up. I will stand by that forever. Anybody else?
I'll talk about mine. I remember probably around the time that the paper that my research is based off was released. I remember my mom telling me about this paper, and it just kind of always stuck around in my mind. And then when I joined my lab and we were talking about olfaction and autism, my PI brought it up, like, yeah, I want to study that. So it's just kind of always been like a long-standing goal for me to be able to study that in our lab.
It's awesome. Yeah, I don't know, any kind of like history that goes into any of this. Like we're all just a collection of our experiences. So it's, yeah, it's interesting to hear from each person about how they got here. Anybody else want to talk about their hypotheses and how?
Sure. I read a lot of papers and so I'm a part of the McNair Scholars Program, and they take you from like grant proposal to finished paper. And it's a lot of work, and part of that initial grant proposal, and also working closely with my mentor. I mean, without her guidance, like the entire summer, because we weren't working in lab, we would have journal club, and we were looking at very vision specific papers and papers that were really relevant to the enrichment project, because my experiments were just a small facet of this larger enrichment project. So we were exploring those papers, and just from lots of reading, from great guidance, it kind of just led to that. I think also I don't want to say, like, obviously with science, it's not good to say it. I mean, it would make sense though, but like the hypothesis, more stimulation, and just given the idea that psychiatrists and pediatricians recommend stimulation, like why would that not apply to the mouse?
Yeah, we're definitely all about model organisms. I don't know. In the classes that I teach, I convince everyone that they're fish. Your fingers are made by the same genes that make fin rays, and so on and so forth. It's just, it is what it is. So all the same questions apply, like most of the time. I appreciate that. Okay, I'm gonna open up a new question. What is the most challenging part of research that you have encountered?
Stopping at the end of the day.
Sometimes that's fair enough. How about like how you came up with your research question, or like you're all amazing people that have made it here. So maybe it's less relevant, but you know, I'm sure that there were hardships along the way.
Writing, like that first step, like now that I've done this a bunch and I've given this talk a number of times, which I did at the end of a very long day today and I stumbled, but now that I've like given this talk a bunch and I've written about it, now it's easy to go up to anybody and talk. But in the very beginning, trying to figure out what to say and what was actually relevant information that I could bring to somebody outside of my field and talk about, that was really hard.
I'll echo that. The visual system is wild and incredibly confusing. And also it just seems so abstract. Like I was talking about spatial and temporal frequency, that took me, I swear, like all summer, and this this book written almost like pop, I suppose, about the visual system for me to like finally grasp. So to be able to explain that in a way to people that aren't familiar with the visual system when it took me so long, it was very difficult. And the presentation you saw today was several edits and presenting to a lot of people who had no idea what I was talking about. And then finally getting to a point where it's just like, my mom was like, okay, I get it. So yeah, communicating to people just in a simple way, just really complex topics.
Anybody else?
I guess like with more of the research central, I think the toughest thing is usually like when you try or you spend like weeks working on an experiment and it just doesn't work, especially with like the Gal4 staining that we do. Sometimes it stains it, sometimes it doesn't, sometimes there's off-target expression. And so I would just say that that's kind of the toughest thing. But yeah, also putting all this stuff into a presentation also is pretty hard.
I think the toughest part for me about research at the moment is not being on campus to conduct the research. I've been remote the entire year, so finally getting to go back this summer will be really great. But it's also taught me in this experience a lot about the analysis that goes on in research, and it's definitely a learning curve, but once you're able to start getting into it, and definitely, Chris Neal's class on MATLAB actually helped me a lot. To learn how to code, especially in MATLAB, which is the program we use for our experiment. But learning these kinds of things have definitely been like the really, really important for me. And also skills I'll definitely take on going forward.
That's awesome. So leading on that, what do you think your next steps are forward for the whole panel?
I would say for me, obviously, first getting back to campus. But as Chris actually kind of pointed out in his question in the Q&A, is actually getting to study the autism mice eventually, once we're able to get through enough mice in the wild type to figure out if this behavior just even exists. Because he asked whether or not ASD mice actually might find different odors positive or negative compared to the wild type mice, if they might just find it completely reversed. And unfortunately, some of the literature, we have not found much literature that's even studied this kind of response in mice. So we have no clue at the moment what will happen in terms of like olfactomotor responses. There has obviously been papers that have looked at attraction and aversion, but I don't know that there have been any that have specifically looked at autism mouse models in response to attraction and aversion, because those experiments are run a little bit differently. They're just put in a box and exposed to the odor, and it's based off of how close or far away they get away to the odor within that box.
I'll be interested to hear if there's a way to leverage things that like don't have an olfactory system. So like take it all the way back down below that, and maybe a lot of it holds true, or maybe a lot of it doesn't. Anyway, same question. Next steps. What do people think?
For research? So for ours, like I said, this was a small component of a larger enrichment project. And because this was exploratory work, like we're not sure if differences will exist. I mean, our results show that differences do exist, but we weren't sure they were going to exist, and we weren't sure at what locations in the brain they were going to exist. So the method that we used for experiments in the project that I presented is a global look at the brain. What's great about the mouse is that their brains are so small, so we're actually able to image the brain, so we can look at all of visual cortex. And that's kind of the first step, because before we could target smaller cell populations, we had to look to see, one, if differences were occurring, and then at what locations those differences are happening. And then, so now that we've seen that there are differences, we have a larger component that's going to be a little bit more behavior-based. I know that there's a goal to do electrophysiology in like freely moving mice. And I know someone mentioned prey capture behavior. We are going to be doing that as well. And the mice are getting an upgrade for their cages, which is fun, which I'm sure they'll really enjoy. And they're going to start hunting crickets. So that's obviously a level up from the food. So it's just kind of moving more towards the more specific behaviors and areas of the brain and cell populations, and trying to figure out like the mechanisms of what's happening.
To second what Alyssa said, so our lab has a new grant proposal to work with Chris Neal's lab so that we can figure out jointly what's going on between auditory and visual cortex. And so, as I'd mentioned, some of our next steps were adding increased noise and then starting electrophysiology data. We've done both of those things actually. And so I'm not doing any experiments though. I'm just doing all of the analysis. But somebody else in our lab has been running the experiments. And so far, I guess they have found that click trains cover the cricket noise better than white noise does. So we've got some data and some video trials from that, and we've got some electrophysiology data. But the problem has been that the electrophysiology data, because of all of the wires that are attached, that creates noise when running deep lab cut. And it creates noise when we run our hidden Markov model, because then our model picks up other things. So there's a lot of trial and error and really going back. So the presentation I gave was fit off of data from the master student that started this project originally, which was a great learning tool of how to use this new algorithm, because our lab had never used a hidden Markov model. And so it was really just kind of like getting our feet wet. But now that we have new data, we have to go through and do these things all over again. So the data I presented used just a single chain hidden Markov model. We've now changed to an autoregressive model, which then has new parameters. And so then everything kind of gets changed and we start the whole process all over again.
Yeah, so for our next steps involving our project, like I said in the presentation, we planned on looking at the hunchback overexpression and knockdowns and how that affects the behavior in MDNs and adult pair ones and MDNs. And we've actually already started looking at that, like I said, in answering the question before. However, I think what I'm pretty excited for is actually the neurite mapping, because the behavioral data that we got, I did not expect to get that kind of behavior. I kind of expected more with the hunchback knockdown for it to just be like a null behavior and look like the vehicle group. And then for like the overexpression for the ATR group to look like the control ATR group. And so it was really interesting actually to see that these were basically exhibiting different behaviors. And so that kind of tells us that these circuitries are being manipulated in ways that we don't fully understand yet. Because like I said before, with the axons, with the knockdown, they weren't able to transcend all the way down the side of the neuropill. So that possibly tells us that they may be able to form like connections with other neurons, or are forming different types of connections with the same premotor neurons. And so I think with the neurite mapping, that would be able to answer a lot of questions for us as to like how these circuitries are being changed when the hunchback overexpression or knockdown is also being changed. And then I think another big step kind of looking after this summer is actually taking a step back and like looking at the spatial transcription factors rather than just the temporal transcription factors. And what these spatial transcription factors do is they're expressed very, very early on in fruit fly development. So these are like in embryos rather than the larvae. And they're a lot harder to research. They don't really like inherit as well as the temporal transcription factors do. However, we have started to look at them, and we're really hoping to figure out which neuroblasts the pair 1s and MDNs derive from. And then if we're able to knock down these spatial transcription factors, how that affects hunchback's binding on the DNA, to see how that possibly affects the outcome of these cells. So yeah, that's kind of our next goals for that project, but yeah.
It goes back to like genetics, and maybe you can find like sequencing assays that will be able to like help you find the chunks that are sticking to different chunks of genomes with cross-linking or something like that, and sequence them. I don't know. It's stuff after my heart.
We actually have a method that's called targeted DAM-ID, where we can actually analyze where hunchback is binding on this DNA by looking at the methylation that it leaves behind using a hybrid protein. But yeah, that's a lot of more biochemistry than most neuroscience labs are used to. But
that's your next talk. It's the next talk. I love it. Okay, I'm going to ask the very, very hard question. Okay, so undergraduate research. If you could go back and start your project over, what would you do differently? Or what would you wish was differently done? Let's go with that.
Start learning how to code earlier. It's so important. It's essential in almost all aspects of research. Like research doesn't exist without coding. So learning that way earlier in my career, I wish I would have done.
I second that 100%. I wish I had learned to code much earlier.
I know it's kind of lame, but I third that.
I actually don't use coding too much in my line of work. I think the only thing that I've really used that has involved coding was the analysis of the behavior, which I was lucky because that was already set up for me basically. But I guess if I could change anything, I guess maybe, I have no idea. It's mainly like procedural stuff and just like knowing which antibodies work better than others, which was kind of something I just had to learn by trial and error. But yeah.
I appreciate all the coding bioinformatician here, again, teaching a bioinformatic master's program. But no, that like Amanda said, like experimental design is like before coding. Incredibly important. So I like hardcore appreciate that. It's the worst thing on the planet when people come to me and are like, here's this data, do something with it. Like, you can't. It's not analyzable. I'm sorry. So no, this is good. I'm really glad that like all of you are thinking about all of these things. That's outstanding. So what do you think is next in your life? You're all finishing up? Halfway finishing up.
I have an extra year, so that's why.
That still counts. Still counts.
Getting there.
So what do you think?
I'm sticking around next year, and I'm going to be working on this project some more. And I'm also working in another lab where I get to use some of the coding experience and data analysis experience that I've gained in this lab to do something different. So I get to explore some other avenues, which I'm excited about.
So I'm graduating, which is really exciting. And I am starting grad school literally the week after I graduate. But I am switching. So I've always been I've always loved research and science communication and education. And so I think whenever I started exploring other classes, I kind of wanted to go more so into science education. So I'm doing a master's program for that and get some experience teaching. And then I hope to go and get my PhD at some point, get more involved in research again and curriculum writing. But for now, it's, yeah, learning how to teach.
Yeah, for me, I still have another year, luckily, because I really like this project that I'm working on. But after that, it's definitely going to be grad school. I definitely want to continue doing research.
Awesome. Okay, we got a couple of random questions in here before we end. What's the biggest aha moment in your research?
Seeing my model do what we hoped and looking at the videos and going, the mouse is in search, and then the model tells us the mouse is in search.
I think for me, I've had multiple aha moments understanding temporal and spatial frequencies. Like I said, they're really confusing topics. So I feel like I get it. And then I'm like, oh, now I get more of it. Okay, now I get it. So I feel like definitely I've had multiple of those and they all revolve around just understanding such a complex sense.
I think for me, with the aha moments, I didn't really realize any of them with like my research, mainly because a lot of this stuff was it just led to more questions after we found out the results. But I think with the learning about the specific experimental techniques and stuff, I think it was when I finally figured out the Gal4 UAS system, because that was definitely a very troubling system to try to understand. But for research, I guess it would be the closest aha moment for that, I guess, would be probably when we figured out that both the MDNs and pair-1s derive from the same temporal transcription factor, because that was a pretty big deal in that this may be the link as to why these neurons know to communicate with each other.
I would probably say most of mine revolves around coding. I remember when I first kind of started out right before the pandemic trying to code a Raspberry Pi camera, and I had no experience. I had very, very little. Like, all right, I'm going off the website, don't know what I'm doing. And I remember going like, yes, when I finally got it to work after like 2 hours of finagling with it. And I took a video of it because I was like, I'm proud, I coded a camera. So definitely like anything revolving around coding or like electrical circuits and building, just to making sure, like seeing it work the way you want it to. So definitely like emphasizing Catherine's point of just when it works.
That's what I'm getting the emphasis is, like, if I got it to work, that's outstanding. And that's, that's a lot of science, in my opinion, regardless of computers or not. So, okay, well, I think we're out of time. So thank you all. Again, one more round of applause. This is an outstanding group of people. So I appreciate you all being here, and thanks for giving us some knowledge. Okay. All right. We'll see y'all later.